Toxicity of drugs remains a significant issue for the pharmaceutical industry, accounting for expensive, late-stage failures in development.
Why is the drug toxicity a significant issue for the pharmaceutical industry? For a host of reasons, really. The first one is that the cost implication of not weeding out compounds sufficiently early, compounds that are doomed to fail for toxicity reason, the cost implications are humongous. It costs $2.6 billion to bring a drug to market. If you have to pull it from the market or even can it in late phase 3, it's very, very costly.
Our track record has been that approximately 1 out of every 10 compounds that's put into clinical development actually makes it to market. So that means our failure rate is 90%, perhaps even higher. So the industry's been troubled by that record for quite some time and is constantly looking for ways to identify compounds that might have toxicity problems, which is the most common reason why compounds fail in development. And so by finding new ways to identify toxicity early, we can streamline the process, we can reduce costs and overhead, shorten timelines, and produce more drugs.
AsedaSciences is developing a sensitive, robust platform to assess toxicity risk in the development of new chemical entities before they become significant financial liabilities.
AsedaSciences is a life science company that has designed cellular-based screening services to help the pharmaceutical industry identify toxicity at the earliest stages of the drug discovery process. One of the challenges that the industry has is the cost of late-stage failures in clinical trials, and the vision for AsedaSciences was to develop a platform by which the industry could improve the overall efficiency by earlier identification of toxicity associated with new compounds early in drug discovery.
Well, the industry has long needed a product that allows them to identify and weed out those compounds that are doomed to fail for toxicity reason. Aseda has that product. It works quickly, and that tox assessment can be done at a fairly modest cost. It's really something that the industry has needed for a long time.
AsedaSciences has developed a platform which is a 3-step process of screen, analyze, visualize. We take compounds early in the drug discovery process, we put them through human cell-based screens, and we look at up to 12 parameters simultaneously that are indicative of cell health. We then take all of the biological information that is produced from these screens, and we have proprietary algorithms that condense that down into a biological fingerprint, and we can then compare that fingerprint to a database of fingerprints from known toxins, withdrawn drugs, and marketed therapeutics. So very efficiently, cost-effectively, we can compare fingerprints and through a process of similarity, identify potential toxins very early in the process and prevent them from going into the drug discovery pipeline.
Flow cytometry is an extremely powerful way to assess the health and well-being of an individual living cell. The technology involves passing cells one at a time through a narrow capillary where they can be irradiated with lasers of specific wavelengths. The cells have been incorporated with markers that, when they light up and fluoresce, can help determine various physiological aspects of the cell, such as mitochondrial function, outer membrane integrity, and the like, features that are generally correlated with potential risk of toxicity down the road.
One of the advantages of early-based screening using cellular models, like the ones developed by AsedaSciences, is that we can prevent these high-risk, potentially toxic compounds from entering into both animal and human trials. And one of the big drives for the industry is to reduce animal testing. Screens developed like the ones at AsedaSciences that can identify these toxicity or toxic types of compounds early in the process prevents those compounds from getting into animal trials where they can potentially fail and cause negative effects in both animals as well as in humans.
The multi-parametric data generated for each compound are then analyzed and visualized using proprietary algorithms to compress the data into a biological fingerprint that is scored based on risk.
AsedaSciences uses our fingerprinting process using a training set of known toxins, withdrawn drugs, as well as marketed therapeutics, and uses those fingerprints to compare to fingerprints of new compounds to assign a level of risk. So the AsedaSciences data identifies risk of toxicity at a much earlier stage than what is currently practiced by the industry. This will allow the medicinal chemists to prioritize which compounds to put forward with less risk and then improve the quality of the drug discovery pipeline.
So the amount of data that can be accumulated in a very short period of time is incredibly rich, and it tells us a lot about how cells are reacting when they're exposed to a test compound. So the assay itself has proven itself to be very robust and very precise and very reproducible. It's, in my opinion, quite unique.
One of the important aspects of developing these types of screens, particularly if you're going to compare fingerprints over time, is reproducibility. AsedaSciences has spent 3 years developing these cellular-based screens so that they're extremely reproducible, and that's not easy to do. So one of the things that AsedaSciences has done has spent a tremendous amount of time and effort to make the overall process reproducible, and that includes the cell culture, and how we grow those cells, how we treat those cells, and how they are used in the assay itself, as well as the automation that's associated with all of the other aspects of the assay, such as adding the dyes that visualize the biological responses that then are used to visualize using the flow cytometry.
The vertical integration of reproducible human cell-based screens, bioinformatics, and cloud computing is an approach that is unique to the pharmaceutical industry.
AsedaSciences is the only company that takes detailed multiparametric biological information, condenses that down automatically into these fingerprints, and then uses those fingerprints to assess risk versus a database of known toxins, withdrawn drugs, marketed therapeutics. That type of vertically integrated approach will be a first for the industry. So by identifying the risk of toxicity earlier, you can improve the quality of the drug discovery pipeline, prevent the later stage failures, and obviously improve the overall safety for patients through that process.
For more information, visit www.asedasciences.com.